Okay, this video is about pack 15. We'll start by looking at cell specialization, how cells join to form tissues and how different tissues are involved in the same function. Organs and then organ systems and then whole organisms. We're also going to think about the differences between growth in plants and animals. And in the last section, we're going to look at a particular behavior of plants where they respond to stimuli in their environment. In particular, we're going to think about how plants respond to light. Go to page 2 of your pack to check the outcomes that we're going to be addressing, and also think about the main objectives of the first part here are to describe how the zygote actually divides by mitosis, and then by the 8-cell stage begins to specialize. And also we're going to think about how those early embryonic stem cells So in this video, we'll go over the key points of how an organism develops, which is pack 15. The first part thinks about specialization of cells and how they join together to form tissues, and how tissues join together to form organs, and organs involved in the same function form organ systems, and then several organ systems form a whole organism. We'll also compare the growth of plants and animals, and in the final bit we're going to look at the particular behavior of plants where they respond to stimuli in their environment, and in particular we're going to look at how they respond to light, which is known as phototropism. As usual, we go to page 2. We study in really, and in the first part of this, we're going to think about cell specialization and what all the stem cells and the development of the multicellular organism. Now, if you think of a single-celled organism, like a bacteria, it carries out all the functions necessary for life. But in a multicellular organism, parts of the organism specialize in particular functions. Probably a more efficient way to do things. But in order to do that, certain cells have to become specialized in their function. And as we saw in an earlier pack, it's really by switching off certain genes. So they only need the genes that they are going to use in their special function. So they switch other genes off. Those genes cannot be switched back on again. So once you become specialized in an animal in particular, that's it. You're in that job forever. Now, we'll see this little picture. We're thinking about a zygote. It divides by mitosis and forms 2 genetically identical cells, and they divide again. And at any point here up to the 8-cell stage, if these cells split apart for some reason, just by accident, can both develop into identical twins. Cell stage, actually, these cells begin to specialize. So ranges of their genes are going to be switched off, and only the genes that they really need, like the heart cell, it's going to have a whole load of genes that encode for, say, proteins that are able to contract. and the will switch, have a load of genes which are involved in transmitting electrical impulses. So as a specialized cell, let's think about zygote, and it divides by mitosis and it produces identical embryonic stem cells. Remember, these cells are not specialized. They can develop into any type of cell. Now, these early stages of development, if these embryonic stem cells separate, they can actually go on to form two completely genetically identical organisms. But by the 8-cell stage, these cells are actually going to begin to specialize. And as they differentiate and become different cells, and as we saw in an earlier pack, the way that happens is certain genes are switched off. And to be a heart cell, I only need the genes which code for the proteins that allow me to do my heart job. For example, I'll have lots of structural proteins that are involved in contracting. That's what makes me a heart cell, the range of genes that I need to do my heart job. I bother to specialize? Well, in a big multicellular organism, I might be a little cell very far away from the outside environment. And in that case, I won't be able to, for example, receive all my oxygen and the food that I need just by diffusion. So I have to have a nice system of transport, like the blood system, specializing in delivering all those nutrients that I need right inside the body. I'm also going to require communication with other parts of the body. And I'm going to require other things too. In a huge multicellular organism, a lot of the cells are not going to be in direct contact with the outside environment. So unlike a bacteria, they're not going to be able to get all their nutrients just by diffusion from the outside. So different parts of the body are actually going to, for example, supply the nutrients to those cells which are deep inside the body. They're going to have to be able to control all that stuff, the oxygen and all the food that comes in from the outside environment. Also going to need communication between different parts of the body. So that's why in a big multicellular organism, we need specialization. Big groups of cells doing particular jobs in order that the whole organism can survive. Nutrients it needs and be able to get rid of all the waste and be able to communicate and interact with the environment. Just a reminder of what cells are, and I recommend you go through this. So use this PowerPoint and do these small tasks. A cell, it's a building block of a living thing. Tissue, what's that? Group of specialized cells involved in a particular function. An organ, more than one tissue. An organ system, more than one organ. An organism, a living thing. Now, up to the 8th cell stage, all the cells in the human embryo are identical. They're embryonic stem cells. They're unspecialized, but they're able to differentiate into any type of cell and, in fact, grow into a whole organism. But from that 8th cell stage, specialization begins. And we're going to need to be able to describe the adaptations of different cells. And we'll be able to see that in the task on page 4 and 5. Descriptions we need. I mean, for instance, the red blood cells, we just need to say that, you know, their function is to carry oxygen. Their adaptation mainly is that they have that shape they've got provides a large surface area so that a lot of oxygen can diffuse into the cell. Now, recall again about the adult stem cells. They're not as versatile as embryonic stem cells, but they're in your bone marrow for the whole of your life, and they are unspecialized, but they can only differentiate and become a certain range of cells, a certain range of blood cells. Now, when groups of specialized cells come together, they form tissues. I think we need to know about 3 main types of tissue. There's epithelial tissue. That's the tissue that basically surrounds organs and it surrounds vessels. There's also glandular tissue. That's formed from the cells that are inside the glands that secrete hormones, such as the liver, the pancreas. Also, muscular tissue is a good example. It's cells that are able to contract, become smaller, and relax, and revert to their original size. And we'll find those in muscles. Also, one of our outcomes deals with the ethical decisions that need to be taken, because we can use embryonic stem cells. They're useful because of their potential to produce any cell, but of course, we have lots of different views about this, and there's many people who think it's unethical to use these embryonic stem cells because that would involve killing an embryo or using an embryo, usually an embryo that has been discarded from IVF treatment. But the outcome makes the point that any work on these embryonic stem cells is subject to government regulation. So in this little activity, nothing. So in theory, we can do this thing called therapeutic cloning, where we can use embryonic stem cells to replace damaged tissues. Again, go to this PowerPoint and use the interactive parts here. We could take a cell from a patient. Remember, it contains all the DNA necessary to turn into any cell, actually. And we can, in theory, put it in a dish. We can then get an egg cell. Remember in our Dolly the sheep cloning, where we took an empty egg cell and we added the nucleus of a body cell to it. The egg cell can then divide and produce stem cells, in theory. And depending on the conditions that we grow these cells in, we grow them on Petri dishes which contain a range of different growth factors and hormones, and we can stimulate the cells to specialize. And in theory, again, we can then perhaps encourage the cells to replace damaged heart cells or damaged kidney cells, and we can put them back in the man and he'll be all happy again. So therapeutic cloning is a potential way to treat diseases such as kidney disease or heart disease. So say we've got a heart disease, there's damage to cells in the heart. We could take a heart cell from the patient. Remember, every cell's got all the genes necessary. And we could use the Dolly the sheep technique for cloning, where we take an egg cell and then we add the DNA from the body cell. Remember, there's two sets of DNA. We use to encourage this fusing of the body cell with the empty egg. And then we could take cells and we grow them, but under different conditions. So they'd be growing with certain hormones and growth factors present. Maybe we can turn these cells, make them specialize into insulin-producing cells you might find in the pancreas. We'd be using this to treat someone with diabetes type 1. We could, of course, treat someone with a condition such as Parkinson's, or maybe someone who's been paralyzed. Cloning, we encourage the specialization of cells into neurons or into heart cells, or anything we like in theory. The other thing, which again is a potential, it certainly doesn't happen yet, but we could grow a whole organ, possibly. That avoids any of the problems of a transplantation, where we've got tissue from another person which might be rejected. Now with plant growth, there's quite a few differences to animal growth. Now, plants, for a start, continue growing throughout the whole of their lifetime. In animals, the growth period is relatively brief. In humans, we grow up to our mid-teens and then we become a full-grown adult. There isn't any further growth. We have replacement of cells, such as skin cells and hair cells and any damaged cells, but the organism doesn't become any bigger. However, plants continue growing for the whole of their life. And the main way they actually increase in size after the cells are divided is that the cells elongate. The cells themselves become bigger. It normally means that they actually become longer. And the actual division of cells only takes place in certain parts of the plant, in places called meristems. Now, meristems are mainly in the growing tips of shoots or the growing tips of roots, but they also find meristem tissue in buds of stems. But that's the only place they actually grow.